start things off.
A massive warm welcome to Coffee Buddies yet again in our marathon activity as an engaging show that occurs once or twice a week, and we call it Coffee Buddies because it's the length of a time to drink a coffee, and it's engaging, entertaining and informing. And as always, it's a great pleasure to see so many of you faces return. And as you know, our format is that we have someone special to come and talk to us, give us a bit of inspiration, give us something different, give us something that we really want to be interested in. And we're lucky to have Bruce to talk to us today. And then after about half an hour, Graham, with his magic touch, moves us around into groups where we can get closer either to Bruce or to people that we don't know and we want to know. And that's the format that we've been working on, and it's working extremely well. And I wanted to really introduce to the group today, it's a great pleasure to have Bruce to come and talk to us, Bruce Feinberg. And Bruce, maybe you could introduce yourself and tell us what you do, please.
Sure. So first, thanks for letting me join your coffee clutch. I can't wait to tell my wife that I'm somebody special.
You're special to us, Bruce.
Yes.
Kind of an interesting, I guess, and different background. I'm trained as a medical oncologist. Did my training at MD Anderson in Houston, Texas, which some of you may be familiar with. Left there in beginning of July of '87 to start a solo practice in Atlanta, Georgia. So I'm from Philadelphia. Thought I'd be returning to the Northeast. My wife's family's more international. She's from Israel. She thought being too close to my parents would be a one-sided kind of relationship with in-laws, and so she wanted to be further away. Had a call to interview in Atlanta, and she loved that as a place to raise a family, and Atlanta turned out to be a great place, not just to raise a family, but start a career. And so my solo practice in cancer medicine turned out to be much more entrepreneurial than I thought I was genetically predisposed to. And so over the course of 23 years, my solo practice grew to a practice with 46 physicians, 36 advanced practice providers, 500 plus staff, 28 locations, 17 separate LLCs. And I was burning out. My original board of directors, all medical oncologists, hematologists, had all gone to what they thought were greener pastures in industry, and I was getting frequent phone calls to join them. But in the summer of 2010, I was feeling that burnout from running this large practice and doing cancer patient care for 25 years, and decided to take up one of them on an offer. So at July 7th, 7/7 at 7 p.m., I took my wife to dinner with my ex-partner and his entrepreneurial mentor to talk about their business that had just been purchased by Cardinal Health. And Cardinal Health was trying to develop a specialty group business. They had been in it before, kind of a false start. They wanted to get more seriously into it. They thought buying some different businesses would be a way to do that. And so I took my wife because never thought I'd do anything else but see patients, and ended up not just seeing patients, but running what became a huge practice, and now leaving that practice and stopping patient care. I was just looking for a soft landing, something else to do, and she was going to do all the talking. We have 4 kids, 2 were in college, 2 were in private school. She was going to sing the sad song of this struggling oncologist who still needed to make quite a bit of income to support this family. She did a great job negotiating. I started at Cardinal September 23rd of 2010, and 10 years later, looking back, it turned out to be a great move. My original work there was actually focused on patient, provider, and payer-facing business units, the most significant of which that they had acquired was a clinical pathways program business. And so the nature of the clinical pathway is the business would end up being the honest broker between a payer and the provider network in that payer in a given specialty, at that point all oncology. And so we would help the network organize, develop a steering committee, and then represent the payer with what their pain points were, and then hopefully develop a series of clinical guidelines that the doctors could live with, at least at an 80% threshold level. And so that business ended up taking off, but I was concerned that I had no proof that this worked. The concept was, if you decrease variance with all these disparate physicians that were out there, that you would have an opportunity to lower costs. So I wanted to look at that. I had done a lot of research in my practice life and while at MD Anderson, and so they gave me an actuary and a PharmD to work with. And by ASCO 2012, I had 4 different opportunities to present what we had done. Kudos to Cardinal. They wanted to actually originally just submit my work, which was positive, as a white paper. And I tried to explain that I'm often confronted by chief medical officers at payers, and doctors don't really think white papers are data. White papers are paid advertising. You just tell the story you want to tell. I said, if we really want to impress the doctors, let me submit this for peer review. And they thought, that sounds neat. And then I explained that if I submit to peer review, the data is going to be embargoed for 5 months. And they weren't so happy about the 5-month embargo of good information. But again, kudos to them. They went along with it. ASCO turned out in 2012 to be a big hit for Cardinal. And this new business that I was managing, this payer pathway business, gave birth to another business, which was real-world evidence research. And for the first 3 years, all the real-world evidence research was being done based on all the claims data that I was receiving from these payer relationships that were driving the pathways, which at that point were 13 different payer contracted pathways.
Just tell us.
But by 2015, it became clear that the pathway movement was migrating in the US. The CMS Innovations Group was looking for a transformation. There was hints they were going to release advanced payment programs like the oncology care model in the US, which would really change the way pathways were done from a top-down payer mandated approach to a bottom-up practice managed approach in which the practices would be doing them in order to improve their profile in a shared risk environment. So we transitioned that program to practices within our GPO, and I then started shifting all my work into real-world evidence research. So that was a really long answer to what sounded like an easy question. I will tell you now, my wife often says that if you ask me the time, I'll tell you how to build a watch. And you can now see, for example.
Well, I think we need to meet your wife and maybe bring her on the show. But I tell you, so, I mean, you're using a lot of language. I mean, we've got an interesting community here, and I'm intrigued whether they understand all the language you use. I mean, this idea, you know, what is real world evidence, and where does it, I mean, if I'm an R&D director in a biotech, or I'm an R&D director in a whatever, I'm an R&D director, why should I be interested in it? Or is it something that the marketing department worries about?
All right, so I'll give you a real life example and the way that I actually got involved in it, because my research had all been clinical trial work. So the year is 1999. As I said, I turned out to be in the right place at the right time. Atlanta turned out to be a booming city. And unlike many of the established metropolises in the US, where there's New York, Chicago, San Francisco, Philadelphia, where you would have 5 or 6 medical schools that would be very much competing for the market share dominance within the region. Atlanta, which now has over 6 million people, had 1 medical school, Emory. They were only graduating 2 fellows a year from the oncology program. And so there was this great opportunity to be entrepreneurial, which I didn't know much about, except that I had instincts. And so as I grew the practice, it grew like a weed. And here I am, even in 1999, we were already 20 plus doctors, and we're able to do a lot of things that were quite progressive and typically done only at an academic center, like doing bone marrow transplant and stem cell transplants and phase 1 and 2 level research. So I'm in this practice. And in 1999, the big story about the first new drug to treat colon cancer in 20 years comes out at the ASCO meeting, the American Society of Clinical Oncology. And if you're a practicing physician, you probably had a lot of colon cancer patients who had been treated with the only drug that was available at the time, 5-fluorouracil or 5-FU, which is a fluoropyrimidine compound, and you've given it every way possible. You've given it daily, you've given it weekly, you've given it over protracted infusions, you've maxed out on the drug, but patients are alive, and you're just hoping they're going to live long enough to get this new drug. And so the drug becomes available, and in my practice then, we already had 20 plus physicians. We all had inventoried patients with advanced colorectal cancer who were going to administer this new drug. And my first patient ends up in the intensive care unit 3 weeks later, highly toxic. Well, I happened to be on call that weekend, and I'm making rounds in the hospital. And when you make rounds in a weekend on call, you're not making rounds on just your patients, but your partner's patients. And next to my patient, literally the bed next to my patient, is my partner's patient in almost the exact same situation, because the drug became available literally on the same day. And many of the doctors in the practice were geared up if they had a patient kind of, we would say, in inventory, hoping they don't survive to see this drug in the same situation. So I come back Monday morning, and we had just implemented our first electronic medical record. And my czar for the medical record, a guy named Richard Lepp, and I said, Rich, you got to help me here. I'm on call. The first 2 patients, I think, who were treated in the practice with this new cocktail on this new, including this new drug, irinotecan, that was the big story at ASCO, both ended up in the ICU, and they both might die. So what's the deal? We should be looking at, with our new electronic medical record, the first 20 patients. Well, we looked at the first 20 patients because everyone, as I said, in the practice was in the same situation. We all had a patient that as soon as that drug became available, we administered to. And so within 1 month of that drug's availability, we learned that of our first 20 patients, 17 were hospitalized, half of them in the intensive care unit. And clearly, something was very different. Because when you look at the clinical trial and the work that's been presented in getting this ballyhoo of activity in the media, both lay media as well as professional media, something's not right. Well, it turns out that the average age in the clinical trial was 62, but the average age in the SEER database, which is the large US database for cancer, was 72. So literally patients are 10 years younger. In the study, only 10% of patients had previously seen that one available drug, 5-FU, yet in my practice, every patient had seen 5-FU and had seen it multiple ways. So that we look at the list of differentiation between the clinical trial and the real world, they were leagues apart. And suddenly it became clear to me that, oh my God, there's like this epiphany. We should be looking at every new drug that comes out. We should look at the first 20 patients in this practice before we just wholesale adopt. And I'm also thinking, wow, if I was in private practice in a solo position, not in this large group where I get the benefit of all this big data, that I'd have this first patient who was a disaster, and then maybe I'd have a patient who didn't do so badly, and maybe the third patient was a disaster, maybe the fourth patient died, and it might have taken me a year or 2 years or longer to understand the reality. And I learned about it literally in 30 days. So we abandoned that new US regimen called IFL and instead used the French regimen, which used these drugs in an infusional way, which diminished their toxicity. And we made that change 4 years before the seminal publication got made in the US that said that that regimen was not ready for prime time. And that the reality of the patients in the community is very different from the highly restricted population that was in the clinical trial.
So you've got this, I mean, one time, you've got it it's written in the label of the drug, it goes out there, and then there's market surveillance, and the market surveillance comes back with a flag that says 70-year-olds get more toxicity. I mean, what practically can you do as a R&D director that moves you towards needing to do a defined population for a clinical trial and understanding real wage data? In other words, is this just something you have to live with? It's a sense and respond. For example, when we get the dreaded COVID vaccine, when we eventually get it, you know, we will want real world data. But I mean, my God, we'll have to wait for years for real world data to feed it back. Where practically can a R&D director fit in real world data into his strategy?
So it's going to be highly variable based on the drug and the disease and the situation. I think the reason that we're seeing a major push, and particularly in the world of cancer medicine, are because of transformations to that patient population in the way that we define them. So we defined back in 1999, we were just beginning to really begin the first real definitional changes. Up until 1985, lung cancer was lung cancer. Then it became small cell lung cancer and non-small cell lung cancer. Then by 1999, we were starting to understand that non-small cell lung cancer was further designed as an adenocarcinoma or a squamous cell carcinoma. Now we go to today. There's over 20 different molecular drivers of lung cancer, each with its own definition. So instead of a population pre-1985 of what would have been 200,000 new diagnoses a year in the US that were all the same, that even when they started to be split, you still had populations of tens of thousands. Now we have populations of lung cancer that are only 1,000 people. How do you start to conduct that clinical trial? And how do you get that information for all this new development that's been helped by genomic medicine and precision medicine initiatives that are creating these small populations, but would take what was 20 years ago a time frame of 12 years to go from bench to bedside with the drug. And you can do various regulatory maneuvers. You can start to do cohort expansion where your phase 2 expands to like a phase 3 without having to initiate a true phase 3 if you have an orphan enough population that's going to be hard to find. But what more can you do? And what real world evidence does is start to say, you know, can't we use a real world population as a comparator? If these patients are hard to find, can't we now do the experimental arm as we do in a clinical trial with all the rigor and all the regulatory requirements? But can't we match that to a real world population and help to drive shorter time to bring it to market, and then maintain phase 4 post-market surveillance for that black box warning or something else that might occur, so we can kind of nip it in the bud before, you know, people might be harmed by something late developing that we wouldn't have seen in too small a population. So I think we have a lot of dynamic changes that are happening now. They're not just happening in cancer. So we're beginning to see that in the world of rheumatology. Rheumatoid arthritis has historically been one disease, one size fits all. But when you talk to clinicians, they will tell you of all these different subtypes within the population that they recognize as bad performers. And yet they didn't have biomarkers that could realistically identify them, but those biomarkers are being found. And so we're going to start to see that in areas of not just cancer and even immunologic disease, but we'll see it across the board as we start to have a better genomic underpinning for what these differences are. And as that defines smaller and smaller populations of patients that we're going to be treating, we're going to need to start thinking about how do we expedite that movement from bench to bedside. And real world evidence becomes a very intriguing way to do that, in addition to regulatory design changes.
So imagine, you know, rheumatoid arthritis is my area. So if you imagine let's think 10 years ahead, Bruce. And I'm an R&D director with an anti-rheumatic. How am I going to be developing it? What's the pathway of development going to look like that brings in real world data?
So I think, again, these are all complex questions, but I'll focus on an area. So what we would typically see is, whatever the drug may be, in most cases, if it's a chronic disease management drug, whether that be a rheumatologic disease or a cancer, that you're going to look at a patient population which has the greatest need, which is usually a patient population that has failed primary therapies. And so you're going to be in that salvage setting. And the reason is it's going to give you fairly rapid endpoints. And there's going to be an unmet need. And you should be able to, again, have a market-to-market strategy that's going to have a shorter timeline. But what if you could actually be looking at a population that is untreated because it has a biomarker differentiation? And so we're starting to see that in looking at ACPA-positive patients in rheumatologic diseases, which is, again, a biomarker that helps to define maybe a bad actor. So I think what we're going to see is a lot of changes that will start to look at how can I define the population of patients. So maybe I can not have to look at a salvage setting, or I can be looking at a salvage setting as well as an early upfront setting. And one of the things that's been dramatic in the world of cancer has been the development of drugs called immune checkpoint inhibitors, drugs made by Merck, Keytruda, Opdivo by BMS, that literally within 5 years have gained 30 indications, are $10 billion plus international blockbusters. And that didn't happen before. It would be 10 years to get your first indication in a salvage setting, not 10 years to have 30 indications, a third of which may be first-line therapy. So I think if you're in R&D space, you're having to think differently. You're starting to think, I can't just think about this disease as a disease. I have to think about what are the subpopulations of this disease? Where are the unmet need within that population? And I think it's a different strategy than you would have employed a decade ago.
But is it something, it's something about these oncology in particular, it's something about the fact that it's smaller populations with, it's all about, you know, taking the empirical disease of a tumor or something, subdividing it up genomically, and you go lower and lower and lower, and then you get this small group, and then what you can do is you can expand, you can understand the real world that that small group lives in. But does this, is this, I mean, are we being skewed by oncology as a thinking partner here? Is there something, is this true for Alzheimer's? Is it true for other things? Is this, you know, and are we really, because oncology is leading the way in the way we do most things actually at the moment. And so my question is there is this all specific to oncology, or is it something that is universally going to be important? The pathways will be rewritten. You won't have phase 1, 2, and 3 anymore. Is that's going to all change?
I think medicine is the pursuit of precision. It's always been the pursuit of precision. No matter what the field is, if we think about, you know, how we examine patients, we've moved from plain imaging to CT scans to MRI to SPECT to PET. We continue to evolve. If you look at pathology, it was straight light microscopy. The problem is very subjective interpretation. Then we started to go into immunohistochemistries. Then we started to move into genomics and understanding the molecular pathways that are governing the process. I think that pursuit of precision is going to be a part of every single disease. We're seeing it unfold in diabetes. We're seeing it unfold in cardiomyopathy. I think it's here to stay, and I think it's going to define. Granted, the marketplace for cancer has been the largest. The amount of money in drug development has been the greatest, so it's been at the forefront of this pursuit. But I don't think it's different in any other field. In the work that I'm doing now, and I'm not just working, you know, whether I'm working in with neuro drugs and MS, with rheumatologic drugs and RA or psoriatic arthritis, inflammatory bowel disease, it seems that that same pursuit is there, in which the biopharma manufacturers are working not only themselves, but with the academic institutions to further understand the molecular and genomic underpinnings of the disease, and starting to look at areas where they can maybe differentiate mechanistically how a drug in their pipeline might be able to express itself.
Do you think, Bruce, if you were an R&D director, you would think, well, what I need to do is reduce the size of my portfolio and spend more money per drug to really get it?
That's interesting. So, you know, going back to my early story, the first thing that I'm always amazed by is you're literally spending tens, hundreds of millions, could be $1 billion to bring a potential blockbuster to the market. And then suddenly you put the brakes on all the research. And I would be researching the hell out of a drug that I release into the market in the first 2 years. I would put a huge amount of my budget to understand if I've already spent X, then to add another 10% on top of X to really understand where the difference is being made. Because often that drug, when it's first released, is being used off-label. It's being used in patients who are much more heavily pretreated than what it was in the clinical trial environment. And then what happens is the provider who's treating that patient has a bad experience and then starts to put a check next to that drug that it's not performing the way they thought, and it was all hype or whatever it may be. And you really want to understand, where is that drug performing in the real world? So I think that I would have different ways of looking at it. Would I focus on a drug and put all my money there? But if I did that, I'd have a huge amount of my budget, I'd have a significant amount of my budget focused on understanding, once that drug is released, how is it really performing? And are there different, is there a different performance in different subgroups? And can I understand them clinically, or can I understand them biochemically or molecularly?
Because the model we've always worked with, I mean, when I worked for Roche, at any one time I had 500 clinical trials operating, but you know, it was a bit like you take it to a certain point and then it goes out, doesn't it? That's what we always did. And what we're talking about is getting much stronger feedback earlier on the complexity of the drug, really, the complexity of the drug working in a disease which is actually much more complex than it ever was, because of the subdivisions that you're talking about, the precision. It's you know, it's now much more complex because it's multiple diseases, isn't it? Each form of genomic form is another disease, and that's bringing complexity in. I'm just mindful of the time, and I'd like to bring some other questions in, because I think this is fascinating. We could talk for ages, but I'm mindful of the time, and I think we'll just open up the chat room, Bruce, and just see whether there's any of our group that would like to ask a couple of questions before we go into the close-up and personal part. Graham, have we got some questions coming in?
Yes, we do. We have Panteli first, and then Kate, and I can see that Tom wants one as well.
Maybe ask Panteli, please. Could you ask your question?
Yeah, no, certainly. So Bruce, thanks for the insight and the feedback, sharing of feedback in that regard. Panteli Theokarous, I'm the cell and gene therapy strategy lead for PPD. So just a quick question around your viewpoint on some of these very innovative therapies being developed in the cell and gene therapy area, and how do we sort of create that balance between, if we consider the financial investments being made around cell and gene therapy and the number of clinical trials that are being conducted, you know, we're seeing that inflection in terms of the projection on some of these things. So how can we sort of introduce that collective approach that's going to ensure that we're bringing these innovative therapies to patients?
Yeah, you're in a much better situation on your side of the pond than we are, because we don't have a healthcare, we have a sick care system, particularly in the U.S. We don't do a very good job at prevention and wellness, and so in our sick care system, it's really been about managing people when they're ill. It's not about really preventing disease or even curing disease. And what cell and gene introduces is the potential for curative therapies. So when you apply an ICER type a process and use a quality, even if you use a quality adjusted life year value that goes back to the original design in 1968 based on dialysis, and you earmark it at that, you know, aged rate of $40,000 a year. You're talking about all of these cell and gene therapies that have the potential to look like, at millions of dollars of cost, a bargain. If you take a patient who otherwise would have 50 years without sight and you give them sight, what's that value? If you talk about a patient who would have had a life expectancy in their 30s or 40s based on a hemoglobinopathy, and they can have now fulfilled their full life expectancy, and without any of the cost or pain for the 30 years or 40 years of that shortened life expectancy. So it really redefines healthcare, and we have to think about, what do we do now? Because everything we've been developing has been about sick care, and now we're talking about truly healthcare and curative intent, not palliative therapy. And you know, there have been a few, you know, situations like in chronic myeloid leukemia where it looked like the drug really may have cured the disease, but for the vast part, whether it's inflammatory diseases of the joints or the bowel, MS, chronic demyelinating diseases or neurodegenerative diseases. I mean, you can pick your area. It's really outside of infectious diseases. We've never really pursued curative intent therapy. And I don't think we really understand how to really develop a financial process to manage it. And it looks to me like it's going to require a public-private partnership. And again, that exists in your side, but not on our side. And I'm not quite sure how we're going to deal with it. And it puts tremendous pressure on our system, and may lead to some very creative financial structures to manage it. But for now, it's what we don't want to do is you don't want to delay or deter the research development, because it really is groundbreaking. And it's not clear, again, what the full extent is. We literally have just a few years under our belt. But it's what's happening right now is pretty impressive.
Yeah.
We've got a few questions in the chat room, so we've got another 5 minutes. If Kate, do you want to go first? Are you there? If you take yourself off mute. You're on
Kate,
mute at the moment.
Yeah, there you go. Hi there. Hi, Bruce. Thanks a lot. It was really just to hear your perspective. We've got a terrible hailstorm here, and all the car alarms are going off. So apologies. It's rare diseases, and I'd love to hear your thoughts on how to approach rare diseases. Is it going to be AI, repurposing drugs, or how do you see us achieving that unmet clinical need? What do you think about that?
Yeah, so it relates to much of what we've been talking about. And so what's interesting is if you think about the original definitions of rare and orphan disease and the numbers that were assigned. In the US, you were orphan if you had, less than, I think the original was 200,000 potential. Because, we were thinking about disease like hypertension and diabetes and, where the numbers were millions and they weren't. So where do you even go? Ultra orphan, and is that going to be under 6,000? But what about diseases that have 1,000 incidences a year, or even less than that? So there are 7,000 hereditary genetic disorders that are potential candidates for cell and gene therapy, and some affect handfuls of patients a year, but they're devastating. It's how do you apply your research dollars? And it's very interesting because one of the things that does happen in the US, which is different, is you really have these 4 distinct stakeholder populations because we don't really have an integrated system. So you've got your payers, providers, patients, and pharma. And interestingly, each of them sees their role with kind of a certain nobility. So obviously patients, the victim, the provider doing the work at the front line, the manufacturer, the government, has control over innovation, but even the payer believes that they're the only adult in the room and they're the steward over the healthcare dollar. Now, it's tainted by the fact that often the steward of the healthcare dollar that they want to own or control rather than really disperse, but they still see a certain nobility in that role. And what it does is it starts to get some interesting dialogue about how do you apply? It may be a fascinating drug for a very small population that has a devastating illness, but if we take those same dollars and apply them to Alzheimer's or cardiomyopathy, we're talking about millions of patients who could benefit. And you start to have to get, put them in line and make your case. So I'm not quite sure, and it does require, again, that kind of a partnership of how do you start to set priorities and what can be done. One way around it is to try to understand, again, biologic mechanisms. And if you can get down to the molecular and genomic level, do you already have things that are out there that might have a role? And we're starting to see that. And for the very first time in the world of cancer, we have cancer drugs that are being approved, not based on a tissue type, but on a molecular aberration. So to think that you have an MSI high and there are a dozen cancers, and based on that microsatellite instability, it's irrelevant whether it's lung cancer, colorectal cancer, similar if it's BRCA positive. And so I think that's one way that I hope to see more research being done, because the cost and time of development is just so profound. If you can do that work at the bench, in the laboratory, there's so much more hope that's potential. Now, that doesn't mean throwing spaghetti at the wall and seeing what sticks.
Okay.
It's a much more educated process than that, although it's the next evolution of throwing spaghetti at the wall and seeing what sticks.
I'm fantastic.
Okay.
I'm mindful of time.
I
Greg.
know, but we're going to take two more questions and then just have two breakouts. So if Tom, if you could go next,
Yeah,
and then we'll follow with Helen.
yeah, Bruce. Thank you. The talk opens up, so many areas, for the future of, genomic research and biomarker, differentiation, which I think obviously is the future. But here's a question for you. If medicine is the pursuit of precision, and we've been lucky, perhaps, with these checkpoint inhibitors and the success of these drugs, when you look at the last count, these, to be a successful checkpoint inhibitor, you're actually hitting, at the last count, something like 18 genes. So how are we going to basically get this matrix of genes which we can target a drug to successfully, when in the past we've just been looking at, say, one receptor gene?
Mm-hmm. Yeah, it's interesting. So, the checkpoint inhibitors were developed in this during the same period, and so we can look at kind of parallel paths of developments between targeted therapies, mostly against tyrosine kinases. So TKIs, tyrosine kinase inhibitors. At the same time, we have immune checkpoint inhibitors, and you can almost look at it as going back to the age-old argument in drug development of lumpers or splitters. Are we going to see this as one disease or one process in which we're going to, in the case of immune checkpoint inhibitors, we're going to arm the immune system of the host, and it's going to be effective against any malignancy within the body? Or are we going to look at every single nuanced potential driver mutation and develop a specific targeted therapy to inhibit that, knowing that that pathway, looks like the Manhattan Metro subway map, and you can get a train's going to get through. You block one line, you block the A train, you can still get to Harlem. And so I think it's a part of that. It's a constant evolutionary argument in how we approach it. So I think when you look at the cell and gene, conceptually, it is one of those more lumper. It's a conceptual approach that says, if we can figure this out, we can do this across multiple disease states. It's similarly with immune checkpoint inhibitors, the first of kind of the host strategies, as opposed to thinking about cancer like a bacteria and an invader strategy. So it's the first of the host strategies, which again has, if it works, it should work against everything. If we can just, I often make it, okay, so I don't know if Star Trek was ever big in the UK, but the Klingons had this cloaking device. And so cancer cells have a cloaking device, and it's more complex than just what they do in their malignant presentation, but they also disarm the immune system. And so if you can just, open up so that the cloaking device is turned off and they can see, the immune system can work. And so I think that there are these kind of driving parallel strategies. And I'm starting to see big pharma kind of make a call. And it's really interesting. So you look at what Merck did, and they decided almost all of their early trials were immune checkpoint inhibitors plus conventional chemotherapy. Whereas BMS wanted to go IO plus IO. And BMS started going first, and it's like horses in the race, and BMS is in the lead. And then the chemo plus is taking the lead, and now Merck has got it. But now IO plus IO is catching up. And so just yesterday, there was a press release with interviews from Merck that, they're still committed to their strategy, but they've got now a half dozen competitors who are using alternative or comparable strategies. And, after BMS had its lead, now Merck took over, and now it looks like Merck is going to lose its lead. So I think we're going to still have this dynamic and these differing pursuits. of how to go after it. But I think in the end, it still comes back to knowledge and understanding mechanism, but not just mechanism of disease, but it's also mechanism of how the host responds to disease. And that's gotten little understanding and getting much more now based on what we've learned in immune checkpoint inhibitor work.
Yeah.
My goodness.
Okay, that's great.
Tom, that's whole body, whole animal
That
pharmacology coming
if we,
back.
yeah.
And I didn't even talk about the microbiome. So there's a lot more to cover here.
Yeah,
Timing,
I think we keep it in the room, Tony. So if we, because there's so few of us, so if we have Helen next on a question, that would
Okay,
be
let's
good.
keep it up. Okay, that's Helen.
So, as an old-fashioned clinical developer, I'm totally with you about the need to get real-world data earlier. But clinical development is already very expensive and very long, and I struggle in many areas to work out how we best add real data early enough to be useful, so we know more about its toxicity and its efficacy, and late enough to be safe, because we're responsible as developers along the way for going stepwise. So it's the dichotomy of what we want versus how we best do it.
So let me give you a real world example, and a real life example. So this is work that I've been doing the past couple of years. So I was engaged, or Cardinal was engaged, but it was kind of my project, by a German biopharma, MorphoSys. And MorphoSys just had their first drug approval, a monoclonal antibody to the cell receptor CD19 on lymphocytes, the drug called tafasitamab, which is going to be branded Monjuvi. Anyway, so they're doing their initial study, and it's a phase 2. And they again, thinking traditionally, they think the drug will have activity in lymphomas, so they want to go to a difficult lymphoma population, which are diffuse large cell lymphomas that are not eligible for bone marrow or stem cell transplant and have relapsed. So this is a pretty small population, and they start to do it, and accruing the patients starts to become a lot harder, and they're watching the clock tick, and they decide that they're going to try to do an expansion study. They get up to 80 patients, but by the time they complete the analysis on the 80 patients, there are now 5 drugs that they are with their drug release. Where they started out, they were all alone. Now they've got 5 competitors and 2 CAR T cells, and 2 other, unique mechanism of action drugs. And it's kind of like their world, is falling apart. And so they have this idea, the only way they can do this, because if they go to a phase 3, they will have lost all their market opportunity. And they're not sure their venture cap is going to support it because of the timeline and a competitive market environment. The only thing they can do is, will the FDA in the US and the regulatory bodies in the EU accept a real-world evidence control arm and accept their expanded phase 2 population in combination as the equivalent of a phase 3? And so that's how I get involved. Can you help us do this? And so they're looking through the literature, what might be convincing to the FDA? And they come up with a 1-to-1 matching population. So the 1-to-1 match is, you literally accrue 10 times the number of real-world patients that you need. So you can literally, from them, distill down that 10% that exactly matches your clinical trial group. And you're matching across 9 covariates. Well, trying to find a population that large was a struggle using many of the conventional sites. We've taken a different approach to real-world evidence, in which we go not to, in the US, the academic institutions, but we actually go to the community practitioners. And they serve as our PIs at their respective sites, and they do the chart abstraction. But as the treating physicians, they can often glean more data than the limited structured data that's in the chart. Bottom line is, we were able to give them 500 patients, which allow them to get to that 1-to-1 matching population, submitted to the FDA, and the FDA thought it was good enough for approval, and they got their drug approved. And so it's a real world, real life example of how real world evidence can start to solve problems. Now, if we go and look at just the FDA as one regulatory body, within the last 12 months, 10 cancer drugs have gained approval based on real world evidence as part of the submission data. Now, that real world evidence may not always be in the label. Sometimes it's kind of the icing on the cake, it's the confirmatory data, but sometimes it's critical. And in this case, it was critical. So I think there are opportunities whereby you can start to think about real world evidence, not just as a post-market surveillance supplement, but rather as complementary data that could even serve, in this purpose of being the control arm. So you don't have to either go to a phase 3, or you can do a phase 3 in which it's half the number of patients you're accruing because you're only accruing to the experimental arm.
Great. I think if we have one last question from Pilar. Be good to hear from you.
Hi, fascinating. Thank you very much. I think you have answered partially to my question. We work with precision medicine companies and in the area of oncology. And even though I'm not expert, what I grasp is part of the research that's going at the moment is about the fact that the drugs themselves change the microenvironment of the tumor along the path. So sometimes it's very difficult to predict how the drug is going to work out for a specific patient. So going on your line of what you said is becoming even more granular, because then it's like with with artificial intelligence and other taxonomies, people are starting to stratify further on. So that means that for some specific cancer groups, there might be very limited commercial value for traditional pharma. So my view is, like, I think you already mentioned that there will have to be some PPP, private-public partnerships. But I just wanted to know how precise or personalized do you think we will go? Because at the moment I think very long trees.
So it's interesting again, and the difference is in the EU versus US. And one of the biggest differences, of course, is the price of drugs. And on average, we're paying 2 or 3 times more in this country. But if you look at that escalation in drug pricing, and you look at it over the last decade or so, it's been an exponential increase. And what that changes, and especially aligns with what we're talking about in this pursuit of precision, is that literally a drug that may only be appropriate for 1,000 patients who will take it for 1 year could hit $1 billion drug in the worldwide or in the US based on pricing. So in the US market, $10,000 a month, per patient for 1,000 patients, and you start to get into ridiculous numbers. So we used to think about, a blockbuster drug is $1 billion drug. You get to have $1 billion drug today with these relatively ultra-orphan populations of patients, if it is going to be a chronic therapy. And so, again, it raises all kinds of challenges, because one, the system can't bear it. I mean, at some point, you're going to have to make really hard decisions. But if we look at the development and why biopharma has, I think, in many cases, been able to pursue, and you could argue that from an innovation standpoint, from these ultra-orphan populations of patients, it's been a huge win, because you can have a population that is that small, but still generate a blockbuster value to pursue it, which historically, often they weren't pursued because it was hard to make the case for all the R&D and the time that would be committed, the effort, the resources for a patient population that small. And so in the old era, it would only be the academic research that would drive it, but that's not the case anymore. So it's kind of weird, because we've got this, contradictions that are inherent to this process, where, the cost is a problem, but the cost also was a huge driver for innovation that benefited an increasing number of ultra-orphan conditions that might not have had that research done. So where do we bring it back together, and how do we come to, to what becomes reasonable and doable. And again, to me, the PPP is the only way to start to sit at how do we prioritize and how do we look at a strategy that is sustainable in the long term and not one that got us where we are with some benefit, but one that we can really pursue for a long-term strategy.
Yeah.
Great. I think, Tony, if you do a roundup now.
Are we at that time?
We are kind of close to it. I mean, if there's a burning question anywhere, put your hand up. We've probably got time for one more.
Questions in the chat room? I don't think there is. I mean, well, fantastic. I mean, we're getting at that time where the coffee is getting cold or the cup is becoming empty. Either way, it's time to finish. And may I just thank Bruce in our traditional way on Coffee Buddies and say, Bruce, fantastic, man. Can you please come back maybe sometime in the new year and tell us how your research is going? Because I think we're all trying to get our heads around real world data, precision medicine, and, you know, what do you spend your money on? Because there's so much to spend your money on. And how the drug, what's the future of the classical way of developing a drug? Is it passé? So we'd like to thank everyone for coming. I mean, Graham, what have we got in the pipe? And just to give everybody an idea.
Yeah, well, we're going through a little bit of a planning stage at the moment, Tony. So, you know, we're looking at doing bigger events going into October. I mean, the next actual event we have is on the 6th of October. We got Eddie Littler from CEO of Reviral, talking about raising his C fund during lockdown. And then we have Kate Arney coming in on October the 8th to talk about her new book.
Fantastic.
So that'll be very, very
we'll
interesting.
Keep you all updated. Please join the app. That's where we're trying to communicate. It's always fab to see you all. And thanks again to Bruce. That was super duper. And we look forward to seeing you again. And be careful out there. And bye for now, folks.
Okay.
Hi, nice meeting you all virtually.
Thank you guys for putting this together.
Thanks, everybody.
Bye-bye.
Bye.